So first, thanks. It's great to be here. I was glad I had an opportunity to talk to you guys tonight. And I thought like this is a good time to talk to you about the work I have because I made an early enough point that it kind of challenged me to get things straight or to get what I think is going on straight in my own mind so that I could try to explain it to others. And so really briefly, what I'm going to be talking about today involves this huge analysis of global forest biodiversity initiative data. And so this is over 200 principal investigators from around the world, and they pool all this forest census data together into one large data set. And then you apply to use this data, and the idea is that we can leverage all of this data to answer some really big questions in biogeography. And it's a good time for the talk because my thinking about this problem has kind of changed from when I was writing the proposal to today. And so when I sat down to write the proposal to use this data, I was kind of in a, that ignorance is bliss, really powerful feeling position. And so I wrote, the story of scientific progress is punctuated by watersheds of conceptual unification, wherein manifold natural phenomena are revealed as end products of a diminishing number of causal laws. And that's a mouthful, but it felt like big and impressive to me. It's this idea that you get a lot of different phenomena, you explain them with an ever diminishing number of laws, and that's science, and that's how we progress. But I kind of got stuck afterwards because I realized that I was thinking really in terms of physics. So like the physics examples of these are like James Clerk Maxwell describing electricity and magnetism in terms of like a unified electromagnetic field. And then you have Einstein's general relativity, and you're taking space, time, energy, and matter, and you're all cramming them into one sort of explanatory framework. And you can kind of see where that's headed. If you keep cramming things into an ever smaller number of things, eventually you end up with just like one grand unified explanation. And this is like our goal in science, or, according to one way of thinking about it. And it's known in philosophy as this idea of monism. There's just one unified reality, and everything we see is just sort of a different aspect of that reality. And if you apply that to science, it's this idea that you have lots of different phenomena, but actually the explanations for them can be diminished down to a point, Pythagorean monad. And at the time when I was writing this, I thought this idea of monism, this idea that we can almost recognize the truth before it comes in the door because we know it will be singular, can be seen as kind of like a guiding light. So, the idea that the continents once fit together into one massive supercontinent, Pangea, predates our understanding of plate tectonics by hundreds of years. And that's because when we see patterns that look like they fit together, our mind naturally jumps to that conclusion. So before we know how continents move, we know they must have been together. We follow that sort of intuitive urging towards sort of a monastic explanation. So at the time when I was writing this, I really felt like I'm standing on the shoulders of giants here. I've got this idea. If you can describe multiple different really big phenomena in terms of an ever diminishing number of rules, you're onto something huge. So without further ado, I want to introduce you to the two things I think, or I thought, were really just one. So the first, this is sort of a tale of two latitudinal gradients. The first is the latitudinal diversity gradient. And this is this idea that tree species richness, and not just tree species, but broadly among lots of different living things, the richness peaks at the equator, sort of these red areas are the highest richness, and then it declines as you move north or south. The second one was this latitudinal mycorrhizal gradient. I'll go into more detail on this, but it's this finding that the dominant form of mycorrhizal fungi, this tree root symbiont that occurs in forests, changes as you move from the equator to either north or south. And so I've colored these things to kind of highlight these two obviously look like they go together, right? There's a clear correlation. I wanted to pause here and give a little background about this mycorrhizal gradient in case we're not as familiar with mycorrhizal fungi. So mycorrhizal fungi, it's this term broadly that refers to a symbiosis between plant roots and soil fungi. And the soil fungi interact symbiotically with the plant root, and they expand the region available for acquiring nutrients. And in return, they get carbon from the plant. and nearly all terrestrial plants associate with mycorrhizal fungi. Now, for the purposes of this talk, there's really only two main functional groups that I'm going to be contrasting. The first is the arbuscular mycorrhizas. This is this group that associates with the vast majority of vascular plants on the planet Earth, and it's thought as the ancestral or the basal state of all terrestrial plants is an arbuscular mycorrhizal fungal one. And in ectomycorrhizas. These are more recent newcomers to the evolutionary scene, but they've been around since about the time of angiosperms, about 100 million years ago. So that 400 million years ago is correct, I see. They associate with a smaller number of plant species, but what's unique about them is that they evolved from these saprotrophic ancestors. That means fungi that are good at decomposing things. And they've retained some of this enzymatic ability to acquire organic nutrients. And that's really the key difference that I want to stress here. Now, if you're looking at forests across the entire globe, you've got something like 70,000 different tree species and tons of different functional traits. But in general, you'd say that about 99% of tree individuals associate exclusively with either the arbuscular or the ectomycorrhizas. So in a sense, if we can add up the proportion of arbuscular mycorrhizal trees in a system plus the proportion of ectomycorrhizas, it equals 1. And we could represent this with just one number, the proportion of ectomycorrhizal trees. It's a simplification, but not a huge one. And that's the one I'm going to be using. So when I talk about mycorrhizal turnover, what I'm saying is the percentage of ectomycorrhizal trees in a system. And if I say a system has 70% ectomycorrhiza, I mean it has 30% arbuscular mycorrhiza. So really briefly, in 2019, I was part of a group where we analyzed this GFBI data, which has got forest census data on over 1 million different sites spread across every forested biome on the planet Earth. We subjected models to random forest training algorithms. So we're looking at trying to predict the proportion of ectomycorrhizal trees in the forest based on different environmental components. And the headline finding of this study was that if we looked at the climatic controls on decomposition rates, so we've got temperature on the x-axis here, precipitation on the y, hot and wet areas tend to decompose leaves pretty fast. Cold and dry areas tend to decompose quite slow. We can use this to create some of these decomposition coefficients. So these are areas that have equal decomposition rate as affected by climate. And if we look at the proportion of ectomycorrhizal trees, what we find is kind of a recapitulation of these classic biome diagrams. So in areas that are hot and wet, we tend to get very few ectomycorrhizal trees. It's mostly arbuscular mycorrhiza. So these are the tropical forests. In areas that are seasonally quite cold and dry, we get a lot of ectomycorrhizal trees. And this is the #1 predictor of the mycorrhizal status of a forest system. So it's kind of like these biomes are structured across this temperature precipitation gradient, and it's determining which symbiotic group is the most abundant. Moreover, if we look at our model's ability to predict mycorrhizal turnover, we find it can recapitulate a 0 to 100% ectomycorrhiza across every continent. So we found an assembly rule that works well everywhere. And so we use this to name a new biogeographic rule called Reed's rule. And it's that climates that slow down decomposition, seasonally cold and dry climates, they favor this tree root symbiosis, the ectomycorrhizal symbiosis, that have advanced decomposition abilities. Climate sets up a barrier. Ectomycorrhizal trees have an ability to crash through that barrier, and that's why they occur in seasonally cold and dry climates. Now, when I was coming up with this and we were doing this analysis, we made these predictive maps of mycorrhizal state, and I was noticing, man, it really lines up well with the latitudinal diversity gradient. These two guys are really strongly correlated, and I started to suspect that there was a causal relationship between them. And this is a different way of looking at it. At the top, you see the I scaled the values, but that dotted line is going to be the diversity, and the solid line is going to be the predicted ectomycorrhizal richness. And then we're going to look at different continents and biomes and see that as you increase the proportion of ectomycorrhizal trees in a system, you decrease the diversity. So I thought they're correlated and they're probably caused. There's probably a causal relationship. And at the time, I sort of told my collaborators about this, and I got some responses that were kind of like this cartoon. They reminded me correlation does not equal causation. I needed to be really careful about what conclusions I made based on correlational data. And I hear that a lot. And what I'd like to reply is that I think that this can be overstated. And in this lecture, I am going to try to tease apart causal relationship with correlational data. But if we're looking at any correlation, let's say between A and B, we find these two are correlated. We've got kind of four different scenarios that create this correlation. The first is that A is causing B. The second is that B is causing A. The third is that there's some confounding variable that's causing both A and B. And the fourth is it's just a coincidence, right? A coincidence like the one that shows a correlation between the number of people who drown in swimming pools and films that Nicolas Cage appears in. So this is from a website devoted to sort of curing people of their tendency to conflate correlation and causation. And so you see it kind of passes the wiggle test. There's a correlation coefficient of 0.66. There's a significant correlation. It looks like when Nicolas Cage appears in more movies, more people die in swimming pools. Now, this comes up a lot. This meme comes up a lot because it's like kind of a cudgel. It's like, oh, you think correlation causes causation? Well, you also probably believe in this ridiculous Nicolas Cage thing. But I kind of took it way too seriously. So when I was thinking about this, I'm like, that's a graph of 11 years of data, right? Nicolas Cage has been making movies for 40 years. And the thing about coincidences is that they're vanishingly rare in really large data sets. So the chances of 11 sort of independently sort of unrelated variables having a correlation that high is about 1 out of every 50. So it's not that huge of a coincidence. But if it persisted through all 40 years, it would be 1 in 300,000. So the only point I wanted to make there, and it's against the people I think who use that correlation doesn't equal causation as kind of a cudgel, is that these sort of coincidences are vanishingly rare in large data sets. And they also affect experiments and descriptive studies alike. So this is why we do things like we correct when we do multiple contrasts. We identify a certain number of questions or comparisons we want to make so that we're not coming up with spurious correlations. And so I would argue, and in this case in particular, that what we're trying actually to do when we look at correlations is figure out whether the causal relationships are indicated by 1, 2, or 3, or some combination of those. And we can sometimes use plausibility, for instance, to distinguish between 1 and 2. We can say, for instance, causes precede effects, or there has to be a plausible mechanism through which a cause can have an effect. But I would just generally make the argument that when we're looking at large data sets and we're trying to tease apart causal relationships, we can come to some conclusions about causality if we control for the correct variables, confounding variables, and we have a good model. That's just my little soapbox thing. So what we have, and what I was looking at, is there's 2 biogeographical gradients. Latitude corresponds to diversity. In this case, diversity declines with latitude. And latitude corresponds with mycorrhizal fungi. So in this case, ectomycorrhizal trees are increasing in abundance with latitude. And what I think is going on is a mediation relationship. So this is where latitude is having some effects on diversity through a non-mycorrhizal pathway, and it's having others that are mediated by mycorrhizal turnover. And what I really want to know is how much of the total effect of latitude on diversity is mediated through this pathway. How much of the effect goes through here versus the top pathway, right? And I say it's almost bound to be some. And the reason why I say it's almost bound to be some is that 85% of all plant species are mycorrhizal. You see, 71% are arbuscular mycorrhiza, and only 2% are ectomycorrhizal. However, if we look at the percentage of tree stems on the planet Earth, 60% of tree stems are ectomycorrhizal versus 40% of arbuscular. Now, if we look at just tree species, so we're eliminating all non-trees from that pie chart, we find that less than 10%, somewhere between, I would say 6 and 10% of tree species form ectomycorrhizal associations. The remaining 90+% form arbuscular mycorrhizal associations. And what we find is that ectomycorrhizal trees are enriched in the most abundant classes. So if we look at the GFBI data set, we've got almost 91 million tree stems total in the entire data set. 28 species account for 50% of those observations. So if in the data set overall, only 7% are ectomycorrhizal. But in this top 28 that accounts for 50%, 20 out of 28 are ectomycorrhizal. So they account for, or they're enriched in these really abundant trees. So when I was thinking about this question, I was thinking kind of in terms of counterfactuals. Let's say you've got 2 trillion ectomycorrhizal trees, and they represent only 7,000 species. Where can we put them and still have a latitudinal tree diversity gradient, right? And as far as I'm concerned, there's really only one answer. We have to put them in the temperate and boreal latitudes. If we allow them into the tropics, they'll swamp out the diversity. We got to put the low diversity ectomycorrhizal trees in higher latitudes and the high diversity arbuscular mycorrhizal trees at the tropical latitudes. Any other alternative assembly rule, an alternative to Reed's rule that allowed ectomycorrhizas to dominate the tropics, would swamp out the latitudinal diversity gradient as we know it. And this is another way of looking at this. Let's say we look at the 7,000 ectomycorrhizal tree species, and we contrast this with estimates of the tree species in the most diverse tropical biome. So this is the moist tropical forests by continent. We find, for instance, that in South America, there's something like 23,000 tree species. So we don't even have 1/3 of that if every ectomycorrhizal tree species occurred in South American moist tropical forests. So that's just to say, we could not fill out a very substantial diversity gradient with just ectomycorrhizal trees. And then I started looking at the GFBI data. Now, what I'm looking at here is a plot of latitude on the x-axis and thousands of tree species. So I just did this. We're looking at, different bands that go all the way around the world. And there's clearly a latitudinal diversity gradient when we look at all species, right? Peaks sharply at the equator and they decline going north and south. And if we look at this, we get kind of a mirror image if we look at both the northern and southern hemisphere. So in most of this talk, I'm going to be talking about absolute latitude. So that's equating -20 degrees and 20 degrees. It's the degree distance from the equator. But if we look at ectomycorrhizal trees, we have to give them a different axis because they're that red line at the bottom there. If we look at ectomycorrhizal trees, they don't have nearly the same kind of latitudinal diversity gradient. In fact, they seem to peak in diversity in the subtropics. And they form this really kind of wicked looking W shape. If we look at these systems in terms of the percentage of species that form the different associations, what we see is that in the tropics, only about 4.5% of species are ectomycorrhiza. And that number increases up to almost 80% in the boreal latitudes. So this is just another way of showing that you do not have enough ectomycorrhizal species to form a strong gradient, and that the only place we can put those arbuscular mycorrhizal tree species in order to sustain a latitudinal diversity gradient is right at the equator, right in those moist forests. There's also Camille Delaveau, who is this PhD who came from the same lab where I did my PhD, and she's gone on to do some incredible work. But she looked at islands as a natural experiment. So what she was looking at was the latitudinal diversity gradient in the mainland on big continents versus oceanic islands. And what you see here is the absolute latitude in degrees and the species richness in terms of trees. And what you see is that on the mainland, that green line, you have a pretty sharp gradient. But on oceanic islands, you have a much flatter gradient. So at the equator, oceanic islands don't have a similar species richness as their corresponding mainlands. And what she found is, by looking at multiple different factors, was that there's this mutualism filter that affects the species richness of islands. So she writes, the mutualism filter strength was the strongest individual predictor of variation. So more so than island size or different environmental variables that she put into her model. She says the species deficit, those are the species that occur in the mainland, but not the corresponding oceanic islands at the same latitude. Species deficit increased where more mainland plant taxa engaged with mutualism with pollinators and fungi and fixing bacteria. In other words, there is a colonization bottleneck. It is disproportionately affecting arbuscular mycorrhizal tree species. Ectomycorrhizal tree species arrive at the island, but arbuscular mycorrhizal species do not. So these oceanic islands filter out the diversity of arbuscular mycorrhizal trees, and they give us kind of a picture of what the diversity gradient would look like without them. And the answer is it would be much diminished. So all these things lead me to believe there has to be a role of mediation. To some extent, the latitudinal diversity gradient has to be mediated by mycorrhizal fungi. The question is just how much of the total percentage, and I wanted to get a percentage. So to do this, I engage in something called causal mediation analysis. So the way this works is first, we look at the effect of latitude on a mycorrhizal mediator. So in the figure on the right here, we've got absolute latitude as our treatment effect. So these models were borrowed from people in the sociological literature. It's not so much used by ecologists, so that's why we're going to have treatment and control effects, even though essentially it's arbitrary which group is which. And what we can see is that if we have an effect of latitude on the mediator, then we can potentially have a mediation effect so long as the mediator then has an effect on our outcome. And what we want to look at is how, when we're talking about the proportion of mediation, is what is the effect of changing the mediator, in this case, changing the mycorrhizal composition while keeping the latitude constant, and how does that effect compare to the total effect of the treatment, or of latitude in this case. So let me try that again. We want to see what happens when we change mycorrhizal fungi but leave latitude constant. That's the effect of changing the mediator. That's our numerator. Then we want to contrast that with the effect of changing both latitude and mycorrhizal fungi. Now in this case, you can see we have two lines. The first line is our predicted latitudinal gradient under the mediator at the control, or the equator. So that would be like an arbuscular mycorrhizal system. This is just example data. And the bottom line would be like if the mediator was at a higher latitude, let's say way up in a boreal forest at 60 degrees latitude. And in this case, what we're seeing is that there's no effect of changing the treatment. This would be if there was just a mediator effect, and we would get a proportion mediation of 1. And if this were the case, we would conclude that the only latitudinal effect on diversity was through mediation. But that's an unlikely outcome. What if we found that treatment has an effect and mediation have an effect? Well, in the case of, for example, a 50% mediation effect, we would see that going from changing the mediator at the equator gives you about half the effect of changing both the mediator and the treatment variable. So changing the mycorrhizal and the latitude gives you double the effect of just changing the mycorrhizal. So this is this idea of mediation. It's saying, what if we just change the mediator variable? What if we just have mycorrhizal turnover? What would this effect be on diversity? And I came up with 4 hypotheses, with 3 given by A and B. And the first one was that latitudinal diversity gradients would be completely unrelated to the mycorrhizal mediator. And when I was thinking about this, I thought this is probably impossible, actually, for the reasons I highlighted earlier. There's just not enough ectomycorrhizal species for there to be no mediation effect. In order for this to occur, it would have to be the case that an ectomycorrhizal tropical forest was just as diverse as an arbuscular mycorrhizal one. And we know that this is not going to be the case because of the differences in their global species pools. The second case was the one I was secretly hoping for. This was the one that was going to explain away the latitudinal diversity gradient just in terms of Reed's rule. This was going to be the case where there's proportion mediation is 1 because there's no effect of latitude independent of mycorrhiza. So I was hoping for this because it would eliminate all of the other myriad mechanisms proposed for the latitudinal diversity gradient, forcing them all through the mechanism that I was a part of, and it would increase my self-importance proportionally. But I knew, even as I did that, that most likely there would be some degree of mediation and some degree of unmediated, or what we would call direct effects. And the difference might be as in terms of whether there's an additive effect of the mediator or an interactive effect. So in this hypothesis 3A, you would get, this would be the example of an additive effect. So mediation would result in the same sort of effect no matter what latitude you were at. Whereas if there's an interactive effect, and this is an example that I put up for a reason, the mediation effect would be particularly large at the equator, but it would be smaller as we got to higher latitudes. Now to test this, I wanted to look at the diversity in different terrestrial ecoregions. So these ecoregions are these areas that have, they're contiguous areas. The WWF, the World Wildlife Fund, has divided the Earth into something like 860 of these units, and they're thought to have similar climate and vegetative characteristics. So I did this to create sort of independent units, and I divide the data set among these ecoregions. And I'm going to go through these panels one at a time. So when you do this, and you've got the top left panel, you're looking at thousands of tree species versus latitude, and each of those points is an ecoregion. And I've got about 801 for which I have data available. And then we can look at the absolute latitude versus thousands of tree species. And the first thing you might notice is that it's kind of messy. When we do just latitudinal bands, we just count every species we get, like we're mowing a lawn, and we can then collect the bag afterwards and look at what's inside. When we do this, we have to combine ecoregions, some of which are diverse, like moist tropical forests, and some of which are more arid and less diverse. And one problem I had when I was looking at the top panels was that the data is not very normally distributed. What you have is a ton of data that have low diversity. Those are those ones, you know, between 0 and 1. They occur at every latitude. And then you have this long tail of high values that seem to occur only at, only within a narrow range around the equator. Now, a classic way to correct for this, sort of so that we can do some statistical analysis, is to take the log number of tree species. And so I log transform this data, and sure enough, you get a much more linear relationship when you're looking at a log tree species. So now we're looking at the proportional effect. We're looking at a multiplicative effect of tree species with latitude. But there's potential issues with this analysis, and one of them is that we needed to correct for the number of observations. And this cropped up in a way that I wasn't expecting when I originally did it. So I did this analysis at first. When you're analyzing things at first, you tend not to correct for everything you should. When I first did this analysis, the first thing I did is I stuck it up on ggplot. So here I have absolute latitude and the log number of tree species. And then I colored my points according to the percentage of ectomycorrhizal trees. And here you can clearly see that there's latitudinal diversity gradient. And as you decrease the number of tree species present at a latitude, you're increasing the ectomycorrhizal. You're going from blue to red across this gradient. But if we look at these points that are ectomycorrhizal, that have a high ectomycorrhizal percentage, they seem actually to occur at a steeper point of the diversity gradient. And on this basis, if you plug this into a simple mediation model without performing any correcting for confounding variables, you would actually expect that a tropical forest that was dominated by ectomycorrhizal trees would have higher diversity than an arbuscular mycorrhizal one. It doesn't make any sense, right? We know that that's impossible. We know that if we planted every single ectomycorrhizal tree on the planet at the tropics, we still would not have a higher diversity gradient. So we know that there's something wrong with these models. And what turns out to be wrong is that the number of observations is too low for these ectomycorrhizal systems. And there's a well-known relationship called the species accumulation curve between the log number of observations and the log number of species observed. Now, if we correct for that, we go from this to this. This is just an example that was useful for me in my thinking about this, is that if you do not correct for the correct, if you do not put in the correct confounding variables, you get qualitatively different predictions that will just be wrong. So there's a big opportunity here for mucking it up, for getting the wrong answer based on the wrong model. Now, this was a clear variable to correct for, but it highlights the importance of getting some of the details of these models correct. And in this case, this model continues to be, this is still a preliminary analysis, it continues to be too simplistic. That said, when we do mediation analysis on this, I arrived at these numbers. There seems to be about a 38% mediation effect at the equator, followed by an 11% mediation effect at 80 degrees, which is the terminus of this data. So that's saying that more than a third of the latitudinal diversity gradient that you see at the equator could be, let's see, the way to say this, going from an arbuscular mycorrhizal to an ectomycorrhizal tropical forest is about equivalent to 1/3 of the total latitudinal species gradient. More than a third of the effect is mediated. And when I found that, I was just crestfallen, actually. I didn't like it at all. That is not the answer I wanted. I tried the analysis so many different ways. You're not supposed to say that, but I did. I was like, this can't be true. So I've done this in many different ways, because what I was secretly hoping for was that we were going to mediate away way more of the latitudinal diversity gradient. Because even 38% mediated suggests that there's about 62% unspecified direct effects. And that would suggest that Reed's rule contributes but does not explain away latitudinal diversity gradients. And yeah, I'm still kind of reeling from that. But I had kind of a revelation while I was trying to talk myself up. And I think that I hit on something kind of true. And I wanted to belabor this point. Throughout this process, I've been emailing with this guy named Mark Lange, because I was reading about philosophy of science. And I felt like lots of people write about philosophy of science, but they do so in a vocabulary using, like terms and things that are difficult for actual scientists to read. But I felt like he was actually onto something. He wrote, lawhood is not something that a fact attains in isolation. Rather, lawhood is achieved only as a member of an integrated set of laws. Research into the mechanisms responsible for various large-scale patterns might fruitfully be guided by the expectation that laws will have to interlock in this way, rather than be utterly independent. What he means is that if we can observe something and sort of promote it to the status of lawhood, it's because it exhibits a kind of stability, a regularity in the real world. And in order for it to exhibit that kind of regularity, it has to be girded up by other forces. It has to be kept under a certain set of conditions under which that law is valid. And I was thinking about this. I'm not sure what happened there. I was thinking about this when I was reading some of the classic literature on the latitudinal diversity gradient. So as I was finding that 38%, which is a big percentage, but not the entirety, was mediated by mycorrhizal turnover, I started to think, all right, what else is there that explains the latitudinal diversity gradient? And in 1962, this guy, or not 62, I think in 1966, Eric Pianka wrote a really influential review, and he came up with 6 different hypotheses for what drives the latitudinal diversity gradient, stating there's obviously room for considerable overlap, meaning they're not necessarily mutually exclusive. We can't line these up and falsify some of them and then only the best survives. It might be that they're working in concert. And I'm like, all right, there's 6 different things. And then I opened up a biogeography textbook, recent one, and I looked at the explanations for the latitudinal diversity gradient. And there's a list with 32 items on it. Unfortunately, this is seen as kind of like an endemic problem in ecology. I think in other fields, other sort of fields that are seen as more soft science as well, is that we kind of explode in ideas, but we don't falsify them. We don't line them up and force them to compete, squid game them, and only the toughest survives. Instead, we let them live on because some of them are catchy, some of them are teaching devices and stuff. And when I saw this list, this is really why I didn't want any of the effect to go unmediated. All of these 32 things could be forced through that unmediated pathway, meaning we would have to wrestle with all of them if we were to make sense of the gradient. And that is not what I wanted. That's not what the Einsteins and the James Clerk Maxwells were doing. They were eliminating things and crushing them together. And I thought, oh, damn it, because now I'm just going to be number 33 on Brown and Lamono, if I even get there. And yeah, so like, I'm not lying. I've been really upset about this. But then, so I had a thought, and I was emailing with Mark Lange. He's a very nice guy in that he immediately responded to my fevered email with lucid philosophical prose. And I was thinking, part of the problem, I think, in my original approach is that I wanted a unified explanation because they're seen as more beautiful and elegant and efficient. But efficiency trades off with redundancy, right? You can't have something that's both efficient and redundant. And redundancy has a function. If we think about big biogeographical patterns, big regularities in the world that we witness as sort of Gulliver and Lilliput, what keeps him pinned down is not one big chord. It's lots of redundant, lots of sort of different mechanisms all pointing in the same direction. What gives us a latitudinal diversity gradient that's persisted over, large epochs of biological time, something that's there to study in the first place, is unlikely to be caused by a single factor that could easily be thrown into some sort of counterfactual situation. In other words, it's unlikely to be driven entirely by mycorrhizal turnover, when mycorrhizal turnover might in itself be affected by climatic instability. The big patterns we see might be those that are tied down redundantly. And I kind of thought about this in terms of a suspension bridge, right? Suspensions bridges are these big, beautiful things, and they don't need that many cables holding them up. Those are redundant supports. And sometimes, if you don't put enough of those cables on the bridges, they collapse. But if we want to bridge the past to the present, maybe we need these bridges with lots of support. So maybe my thinking about it was wrong. So maybe I should be pleased to have found some mediated effect and to be okay with being part of the story, but not the dominant factor in the latitudinal diversity gradient. Anyways, that's what I've been telling myself. And this work involves, it's not finished. This is preliminary work. I have a lot more analyses I'd like to do on it, but this is work I do in collaboration with a huge number of people. And the only reason I can play around with this data is that a huge number of people collected it. They literally marked and tagged it. 91 million tree stems, and the infrastructure and everything that goes into collating this data is enormous. So just want to thank my collaborators.